I'm really embarrassed because I only watched one Ignite video and it wasn't particularly funny, so my presentation is not particularly funny, but I'll get on with it anyway. Synthetic biology is a new emerging technology. Craig Venter pioneered it 3 years ago with the transfer of a complete bacterial genome from one organism to another after he'd written it and edited it on a computer. And he said, over the next 20 years, synthetic genomics is going to become the standard for making anything. I look forward to the time when I can look back 20 years and do a talk like Jen just did. He has a company called Synthetic Genomics, but it's not the only game in town. It was a few years ago, but the number of companies has been increasing exponentially. And we ran an incubator for these companies a few years ago and found that there's this actual underlying ecosystem of synthetic biology startups. The reason why the number of companies are increasing is because it's becoming increasingly important in the US economy. $300 billion, or 2% of GDP, in genetically modified stuff, roughly split between crops, drugs, and chemicals. Here are a selection of some of the companies. Some of them are here at FU, including Cambrian Genomics, who are doing DNA laser printing. There are different kinds of companies, and I'm going to go into some of the different ones. We ran the first SYNBIOBETA conference in in Menlo Park last year, and we've got another one coming up in San Francisco where we brought together members of this nascent startup community. The different areas, there's we call the SynBio stack. Gene and genome synthesis, that's like the stuff that these organisms are made of. CAD tools, computational design tools for designing genes and genomes. Organism engineering platforms. And finally, the applications, what the companies are actually selling. DNA comes in many different sizes. Oligos are maybe 100 A's, C's, T's, and G's, and it could just cost you $6. You could order them today and get them in the mail tomorrow. There are different technologies building up to whole genomes, and it's about a $250 million market. Carlson's curve is our equivalent of Moore's law, and Rob Carlson plotted it here on a log scale, the falling cost of sequencing. Now your whole genome could be sequenced just for a few $1,000s, and you can now synthesize an organism for about $1 million. It's halving every 18 months. A bunch of different companies making different lengths and with different technologies, and some next generation companies at the end there and what they're working on. Next up we have CAD tools. So great, you can synthesize 1 million base pairs, but what does it do and how do you organize that information? So computer-aided design tools for genes and genomes is a big area, and it's fairly low barrier to entry, so we got quite a few companies getting in on the game. Genome Compiler you may have heard of. They did the glowing plants on Kickstarter recently. Autodesk is a big player in this area. It really sees the bioprogrammable matter as a big deal. DNA 2.0 down in Menlo Park. This is a screenshot from teselagen Biotechnology, which came out of JBay, Joint Bioenergy Institute in Emeryville. And here's a screenshot of their program, which is a drag-and-drop interface for genes and genome design. On the next level up, we have organism engineering platforms. These are the APIs and the tools and tool makers that make it easier to engineer biology. So they're making sets of switches or sets of technologies for directed evolution that make it easier to engineer and make other applications. Some of those companies are shown here. Synthetic Genomics is one of them. That's Craig Venter's company. Ginkgo Bioworks over in Cambridge, or in Boston rather, and Genomatica down in San Diego. So finally, the applications. What are we going to do with this cool new technology that we have? Well, there are a number of different areas of application. One of them, the most common, is biofuels. That's because the federal government has dumped hundreds of millions of dollars into sustainable fuels. And you might recognize some of these if you're in the Bay Area. Codexis, LS9, Amyris is over in Emeryville as well. So most of them started off in biofuels, but anything that's made petrochemically, in general, you can make it cheaper, faster, and more sustainable through biotechnology. Here's a case study of Amyris. They take in plant sugars from sugarcane in Brazil. They have an industrial platform for engineering yeast cells to ferment these sugars into higher value chemicals. Here's my favorite example of all. In San Francisco, Refactored Materials have taken the genes from the spider to make spider silk. They've engineered them into E. coli, and now in a fermenter, instead of making alcohol, you can make spider silk. If none of this made sense to you and you don't know how a gene relates to a protein, or you've never taken a biology class since high school, we run a course, Synthetic Biology for Computer Programmers. And if you're a friend of FU, then you're welcome to send me an email and come to the course for free. Thank you all.